Introduction to Biochemistry. Biochemistry is the branch of science that explores the chemical processes within and related to living organisms. It is a multidisciplinary field that combines biology and chemistry to understand the molecular mechanisms that sustain life. This presentation provides an overview of biochemistry, focusing on the fundamental metabolic processes and the vital molecules that drive biological functions. Key topics include microscope biomolecules, understanding carbohydrates, proteins, lipids, and nucleic acids, the essential building blocks of life. Gear metabolism, exploring catabolic and anabolic pathways, how the body converts food into energy, and the role of enzymes in biochemical reactions. Counterclockwise arrows button cellular energy, examining ATP production, glycolysis, the Krebs cycle, and oxidative phosphorylation. DNA genetic biochemistry, studying DNA, RNA, and protein synthesis, which are crucial for heredity and cellular function. By delving into these topics, we gain insight into how biochemical processes regulate life at the molecular level and contribute to health, disease, and biotechnology. Rocket. Metabolism, anabolic and catabolic processes. Metabolism refers to the total sum of all biochemical reactions occurring in a living organism to maintain life. These processes are broadly classified into anabolic and catabolic pathways, which work together to regulate energy flow and maintain homeostasis. 1. Anabolic processes, biosynthesis. Definition. Anabolism is the set of metabolic pathways that construct complex molecules from simpler ones, requiring energy input. Energy requirement. These processes are endergonic, meaning they consume energy, usually in the form of ATP. Functions. Building macromolecules like proteins, nucleic acids, and lipids. Tissue growth and repair. Storage of energy in the form of glycogen or fat. Examples. Protein synthesis. Amino acids, proteins. DNA replication. Nucleotides, DNA. Glycogenesis. Glucose, glycogen. Lipogenesis. Fatty acids, triglycerides. 2. Catabolic processes, breakdown. Definition. Catabolism refers to metabolic pathways that break down complex molecules into simpler ones, releasing energy. Energy release. These processes are exergonic, meaning they release energy that is used to generate ATP. Functions. Providing energy for cellular activities. Recycling cellular components. Eliminating waste products. Examples. Glycolysis. Glucose pyruvate plus ATP. Krebs cycle, citric acid cycle, breakdown of acetyl-CoA to produce ATP and electron carriers. Lipolysis. Breakdown of triglycerides into fatty acids. Proteolysis. Breakdown of proteins into amino acids. Anabolism versus catabolism. Key differences. Purpose. Anabolism is responsible for building complex molecules from simpler ones. Catabolism, on the other hand, breaks down complex molecules into simpler forms. Energy usage. Anabolic processes require energy, usually in the form of ATP. Catabolic processes release energy, which is often used to generate ATP. Process type. Anabolism is associated with biosynthesis, where new cellular components are formed. Catabolism involves the degradation of molecules to extract energy. Examples. Common examples of anabolism include protein synthesis, DNA replication, and glycogenesis, conversion of glucose into glycogen for storage. Examples of catabolism include glycolysis, breakdown of glucose for energy, lipolysis, breakdown of fats, and cellular respiration, energy production through oxidation of molecules. Conclusion. Anabolic and catabolic pathways are interconnected, forming a balanced metabolic network. The energy released from catabolism is used to power anabolic processes, ensuring the continuous functioning of cells and organisms. This balance is crucial for growth, repair, and maintaining overall health. Insulin and glucagon, the dual regulators of metabolic homeostasis. Insulin and glucagon, two antagonistic hormones secreted by the pancreatic islets of Langerhans, play a pivotal role in maintaining glucose homeostasis and orchestrating metabolic balance. Their finely tuned interplay regulates the flux of nutrients across various biochemical pathways, ensuring cellular energy demands are met while preventing metabolic cells of the pancreas in response to hyperglycemia, elevated blood glucose levels. Metabolic role. Insulin functions as a key anabolic hormone that promotes macromolecule biosynthesis and energy storage. Mechanisms of action. Glucose uptake and utilization. Upregulates GLUT4 translocation to the plasma membrane in insulin-sensitive tissues, muscle and adipose, facilitating glucose influx. Enhances glycolytic flux by stimulating hexokinase and phosphofructokinase activity. Glycogenesis and lipogenesis. Induces hepatic glycogen synthesis via activation of glycogen synthase and inhibition of glycogen phosphorylase. Stimulates de novo lipogenesis by upregulating acetyl-CoA carboxylase and fatty acid synthase. Inhibits lipolysis by suppressing hormone-sensitive lipase, H-cell. Protein anabolism and cellular growth. Activates mTOR signaling, enhancing ribosomal biogenesis and protein translation. Suppresses proteolysis via inhibition of ubiquitin proteasome pathway, the catabolic counterbalance. Secreted by alpha symbol, cells of the pancreas in response to hypoglycemia, low blood glucose levels. Metabolic role. Glucagon serves as a catabolic agent that mobilizes energy reserves, ensuring glucose availability during fasting or metabolic stress. Mechanisms of action. Hepatic glycogenolysis and gluconeogenesis. Activates adenylate cyclase MPK signaling to phosphorylate and activate glycogen phosphorylase, promoting glycogen breakdown. Enhances gluconeogenic pathways via transcriptional upregulation of PPCK, fructose 1,6-bisphosphatase and glucose 6-phosphatase. Lipolysis and ketogenesis. Stimulates adipocyte HSL, increasing free fatty acid mobilization. Augments hepatic beta oxidation, shifting metabolic preference towards ketone body synthesis under prolonged fasting conditions. Amino acid catabolism. Increases hepatic transamination reactions, facilitating gluconeogenesis from amino acid substrates. Insulin-glucagon axis. Dynamic metabolic equilibrium. The reciprocal regulation of insulin and glucagon maintains glucose homeostasis through a tightly controlled feedback loop. During the fed state, insulin predominates, directing glucose towards storage and anabolic processes. In contrast, during the fasted state, glucagon dominates, liberating stored substrates to sustain energy demands. Pathophysiological implications. Diabetes mellitus. Type 1 diabetes, T1D. Results from autoimmune destruction of beta cells, leading to absolute insulin deficiency. Type 2 diabetes, T2D, arises from insulin resistance and beta cell dysfunction, disrupting metabolic homeostasis. Hyperinsulinemia and insulin resistance. Chronic hyperinsulinemia can desensitize insulin receptors, leading to compensatory hyperglycemia. Glucagon dysregulation. Excessive glucagon secretion as observed in diabetes and certain pancreatic tumors, glucagenomas, exacerbates hyperglycemia by unchecked hepatic glucose production. Conclusion. The intricate balance between insulin and glucagon is essential for metabolic stability, governing glucose availability, energy partitioning and cellular homeostasis. Any perturbation in their signaling dynamics can precipitate metabolic disorders, underscoring their critical role in endocrinology and systemic physiology. Amino acids, building blocks of life, protein synthesis, enzymes and hormones. Amino acids are organic molecules that serve as the fundamental building blocks of proteins, which are essential for the structure and function of living organisms. These compounds contain an amino group, NH2, a carboxyl group, COH, and a unique side chain, R group, that determines their properties. 1. Role in protein synthesis. Protein synthesis is a biological process in which amino acids are linked together by peptide bonds to form proteins. This process occurs in two main stages. Transcription. DNA is transcribed into mRNA, which carries genetic instructions. Translation. Ribosomes read the mRNA sequence and assemble amino acids into a polypeptide chain with the help of tRNA. Proteins formed through this process are crucial for cell structure, function, and regulation. 2. Amino acids and enzymes. Enzymes are specialized proteins that act as biological catalysts, accelerating chemical reactions within cells. They are composed of specific amino acids that form the active site, where substrate binding and conversion occur. Without amino acids, enzymes would not be able to function, affecting metabolism and other vital biochemical processes. 3. Amino acids and hormones. Many hormones are peptides or proteins synthesized from amino acids. For example, insulin regulates blood sugar levels, is a peptide hormone made of amino acids. Thyroxine, T4, is derived from tyrosine and controls metabolism. Epinephrine is another amino acid-derived hormone that plays a role in the fight-or-flight response. Amino acids are indispensable for life, contributing to growth, repair, immune function, and overall homeostasis. They ensure the proper functioning of proteins, enzymes, and hormones, making them the essential building blocks of life. The role of ATP in metabolic pathways: energy currency transfer and metabolic coupling. Adenosine triphosphate ATP is the primary energy carrier in biological systems. It plays a crucial role in metabolic pathways by serving as an energy currency, facilitating energy transfer, and enabling metabolic coupling to drive essential cellular processes. 1. ATP as the energy currency of the cell. ATP acts as the universal energy currency because it stores energy in high-energy phosphate bonds between its 3 phosphate groups. The hydrolysis of ATP into ADP, adenosine diphosphate, and inorganic phosphate, Pi, releases approximately 7.3 kilocalories per mole of energy, which fuels cellular activities. ATP is continuously regenerated through cellular respiration and other metabolic processes to sustain life. 2. ATP in energy transfer. ATP plays a vital role in transferring energy within cells. Phosphorylation. ATP donates a phosphate group to molecules, e.g., glucose phosphorylation in glycolysis, activating or deactivating enzymes and metabolic intermediates. Mechanical work. ATP fuels muscle contraction, intracellular transport, and cytoskeletal rearrangements. Chemical work. ATP drives biosynthetic reactions, e.g., protein and DNA synthesis. Active transport. ATP is required for ion pumps like the Na+/K+ ATPase, which maintains cellular homeostasis. 3. ATP and metabolic coupling. Metabolic pathways are interconnected through ATP. Catabolic reactions, energy-producing processes like glycolysis, the Krebs cycle, and oxidative phosphorylation generate ATP by breaking down macronutrients, carbohydrates, lipids, proteins. Anabolic reactions, energy-consuming ATP provides energy for biosynthesis, such as protein synthesis and lipid formation. ATP-regulated enzyme activity. ATP modulates key metabolic enzymes, ensuring energy balance and efficiency in pathways. Conclusion. ATP serves as the central molecule in metabolism, efficiently linking energy generation to energy-demanding processes. Its role in energy storage, transfer, and metabolic coupling makes it indispensable for cellular function and survival. Maintaining metabolic balance. Homeostasis, regulation, feedback loops, and dynamic equilibrium. The human body is a complex system that constantly works to maintain a stable internal environment despite changes in external conditions. This process is called homeostasis, and it is crucial for the proper functioning of all biological systems. Homeostasis involves several mechanisms of regulation, feedback loops, and dynamic equilibrium to maintain balance in various physiological processes. 1. Homeostasis. Homeostasis refers to the body's ability to maintain a relatively stable internal environment, despite fluctuations in the external environment. This includes regulating factors such as body temperature, blood pressure, blood glucose levels, and the concentration of ions, like sodium, potassium, etc. For instance, the body keeps the internal temperature around 37 degrees Celsius (98.6 degrees Fahrenheit) through mechanisms like sweating or shivering. The body regulates blood glucose levels to provide consistent energy to cells, typically around 70 to 110 mg/dl. 2. Regulation. Regulation is the process through which the body maintains homeostasis. It involves detecting changes from a set point, optimal value, and initiating appropriate responses to correct the imbalance. For example, temperature regulation. If the body temperature rises above the optimal range, sensors in the skin and brain detect the change, and mechanisms like sweating to cool down, and vasodilation, widening blood vessels to release heat, are activated. Blood glucose regulation. When blood glucose levels rise after eating, the pancreas secretes insulin, which helps cells take in glucose, lowering blood sugar levels. Conversely, when blood sugar drops, glucagon is released to stimulate the liver to release glucose into the bloodstream. 3. Feedback loops. Feedback loops are the mechanisms that regulate homeostasis. There are two main types. Negative feedback. This is the most common feedback mechanism in the body. It works to counteract any changes, bringing the system back to its set point. Negative feedback loops help maintain stability. For instance, if blood sugar levels rise, insulin is released to lower them. When the levels drop too low, glucagon is released to increase them. Example. Temperature regulation is another example of negative feedback, where the body cools down or heats up depending on the surrounding environment to maintain the set temperature. Positive feedback. This feedback mechanism amplifies or increases a response in the body, moving further from the set point. Positive feedback is less common than negative feedback and is typically used in processes that need to be completed quickly and decisively. Example. Childbirth is a classic example of positive feedback. During labor, uterine contractions stimulate the release of oxytocin, which intensifies contractions, which in turn causes more oxytocin to be released, until the baby is born. 4. Dynamic equilibrium. Dynamic equilibrium refers to the state where physiological systems continuously adjust and stabilize to maintain homeostasis. While the internal environment is in constant flux, it stays within a narrow range of optimal values. These constant adjustments ensure that the body remains functional despite external changes. For example, breathing. The body continuously adjusts breathing rate based on oxygen and carbon dioxide levels in the blood. When CO2 rises, the body responds by increasing the breathing rate to expel excess CO2 and bring in oxygen. pH regulation. The body maintains blood pH within a narrow range, 7.35 to 7.45. If the blood becomes too acidic or too alkaline, mechanisms involving the kidneys and respiratory system adjust to bring it back to equilibrium. Conclusion. In summary, maintaining metabolic balance is vital for the body's survival. Through homeostasis, regulation, feedback loops, and dynamic equilibrium, the body can adapt to internal and external changes while keeping essential physiological variables within an optimal range. This constant balancing act allows the body to function efficiently and stay healthy. Introduction to Biochemistry. Welcome to this insightful presentation on biochemistry, where we explore the fundamental metabolic processes and the vital molecules that sustain life. This session aims to provide a clear understanding of key biochemical principles and their significance in various biological functions. The PowerPoint version of this lecture is available upon request for further study and reference. This lecture has been carefully designed using advanced AI tools and is proudly presented by Ghulam Farooq, CEO of AI Tools Port. Our goal is to make complex scientific concepts more accessible and engaging for learners worldwide. If you find this content valuable, we would truly appreciate your support. Please like, share, and subscribe to our channel, and don't forget to hit the bell icon to stay updated with our latest educational content. We welcome your questions, thoughts, and feedback in the comment section below. Your engagement helps us improve and bring more informative lectures to you. Thank you for being a part of our learning community.